Hello and good morning. So here we are. We are looking at our current lab for the first week of January. I believe for us, this is going to be lab, is it 18 now? No, whoa, we're way past that. Lab 23, the Monster Genetics Lab. So let me show you and explain it. Now, we are going to split this into part 1, part 2, and part 3. But I am going to explain the whole lab in this video so that I don't have to make 3 videos on it. So here is our Monster Genetics Lab. Now, basically, what this lab is going to do is it's going to be our final review for the patterns of inheritance before we move on and we start getting into actual DNA genetics. And if we take a look, it says you've learned about the different patterns of inheritance. We covered that today. Some simple dominance, others like incomplete and codominance. So now we're going to use all of them. Our procedure. Number 1, flip a coin twice to determine the genotype for each trait and record it in the data table. Heads on the coin means allele 1. That's this column right here. You can see it says allele 1. And I highlight allele 1 and allele 1. And then tails will be allele 2. And here is the column for allele number 2. Example, if you flipped heads twice, your monster would have 2 copies of allele 1 for his genotype. Number 2, determine the phenotype resulting. You're going to look right here in the table. And then repeat steps 1 and 2 for all of these different traits. It's really not too bad. So let's take an example. All right. So I have my coins. I look at the trait for eyes. All right. Allele 1 is 2 small eyes, capital E. Allele 2 is 1 large I, little e. So which one is dominant? Allele 1 is, because it's capital. Which one is recessive? Allele 2 is, because it's lowercase. Now I take my 2 coins, flip them. I got 1 heads and 1 tails. So heads is allele 1 right here, 2 small eyes. And a tails would be 1 large I, little e. So the genotype I'm going to get is 1 capital E from the first column, 1 little e from the second. Now, what phenotype is that? What physical trait? Well, we look back here. Capital E is dominant over little e, so it's going to have 2 small eyes because the dominant one wins. All right. Now we move on to the next. Now this one for eye color. This tells me this is incomplete dominance. So we have 2. We have R, capital R, which is dominant for red. And then we have capital R dash or capital R prime, which is also dominant for white. Okay, well, we still follow the same rule. Take your coins. And I got heads. So that means that I get allele number 1. So I get a capital R and a capital R. What phenotype do I have? Well, 2 capital R's, that's red. There's nothing else mixed in there. Now, let's imagine, though. Let's imagine that you got capital R and then R prime. So you got 1 of each. This is incomplete dominance. Remember, incomplete dominance means blending. So what do we get when we blend red and white? I'll have to take red paint and white paint. You mix them together, you get pink. Right? Blended. Skin color. This one is codominant. So we've got capital G, dominant for green, and capital B, dominant for blue. Do the same thing. Take your coins. Now I got 1 heads and 1 tails. So I am going to get a capital G, capital B, 1 of each. And because it's codominant, remember, codominant means you get both of them together at the same time. No difference. They're both going to show up. So its phenotype is going to be green and blue. Because it'll show that maybe it's striped, maybe it's spotted. It doesn't matter. Maybe it has, like, I don't know, blue hands and a green body. Whatever, that's fine. It's just showing both green and blue. You're going to repeat this process all the way through. Now, people may get confused with tail and ears regulatory. All I can tell you guys for that is don't worry too much about it, because you see you're going to get alleles for tail shape and tail color. All it matters when we say regulatory is, do you have it or not? So if you get capital T, your monster will have a tail. But if you get 2 lowercase t's, then you have no tail. You have the genes, you have the DNA to make a tail, but your tail is just turned off. That the regulatory switch was turned off, so you never produce it. To put this in perspective, human beings, we still have tail DNA in our genetic code from our simian ancestors hundreds of thousands of years ago. It's still there. We just don't use it. Our regulatory DNA just is flicked off. No need for it. Not currently. So, part 1 is what I just showed you, completing this data table on the first page. That is our goal, Tuesday, January 5th. Part 2 is figuring out this data table. Now, this goes very quickly once you understand it. You're going to be flipping back to this data table in order to help with what traits and what letters mean what. So now we're looking at the phenotype of a male monster and its genotype. We need to work back and forth. So what we need to do is we need to take a look and go, eyes. Okay, well, this monster has little e, little e. So we come back up here. He has 2 little e's. So that's 2 recessive alleles for 1 large eye. So his phenotype will be, 1 large eye. His eye color is white. Okay, we come up here. Eye color, capital R is red, capital R prime is white, and they're incomplete. So if I have one of each, they're going to blend and make pink. So the only genotype he can be and have a white eye is going to be R prime, right? He has to be homozygous for it. Skin color. All right, this is our codominant one. We come up here. Capital G is green. Capital B is blue. So if you have 2 G's, you're green. If you have 2 B's, you're blue. And if you have 1 of each, you're both green and blue. Well, he's just green. So he's got to be capital GG. Right? And you guys are just going to go through and finish out the rest of this data table. Nice, easy, simple. That's part 2 for Wednesday. Some of you will blow through this in 5 minutes. Other people will find it more challenging, and you'll need a little bit more time to wrap your brain around how this works. But we have that time. Not a problem. Part 3. Part 3 we will be doing on Thursday, January 7th. And now we're going to come down and we're going to make Punnett squares. Now, you're going to need to attach your work because I want to see all 8 of these Punnett squares. You're going to take your 2 monsters, the female monster that you flipped for in part 1 on Tuesday, the male monster that you figured out on Wednesday. We have both of their genotypes here and here, and now you're going to make Punnett squares for each one of these crosses for eyes, eye color, skin color, tail, feet, horn, ears, claws. And you're going to answer your percentages for each one. So, for example, this is going to look terrible because I'm just going to try to draw it freehand. I'm just going to put it right here. Right? There's our terrible box. Let's imagine we do the first one. Eyes. All right. Our girl monster in this example was big E, little e. So I'll take hers and I'll spread them out because of segregation of alleles. Our male monster is little e, little e. And now we complete the Punnett square. This is really unpleasant with the mouse. All right. We've completed the Punnett square. What percentage of your offspring will have only one eye? Well, in order to have one large eye, they must be homozygous recessive. And that's going to be 1, 2, 2 boxes in this example is going to be our, oh, that's a terrible 50. Will be 50%. And you'll do that for each one of these crosses. So, ladies and gentlemen, that is our Monster Genetics Lab. Hopefully that will help everybody kind of see their way through it. I'll be here in class. And this also gives you a little bit of extra time this week to, once again, finish up work that may need to be done, get done with labs that may not yet be complete. Ladies and gentlemen, have an excellent day. And I look forward to seeing everybody in class, as always.